Okay, so for this video, we're going to be talking about the idea of annotating cell types. And so previously, Jesse had gone over how when we take single cell data, obviously we can't look at every single individual cell and understand exactly what it's doing. We need to form more meta-analyses to get a good understanding of what the cells are doing, while also taking advantage of the fact that we have single cell data. And so the way we can do this is we can cluster cells together that we expect are acting similarly. But after that, we need to get an idea of what is causing these differences and what do we already know in the literature that can give us good information about what these cells are doing and their defined importance. And that's where annotating the clusters as certain cell states or cell types is essential. And there are two kind of key ways that we can do this. One is using an already curated atlas. So single cell is still new, but there have been a lot of efforts into making atlases, or basically people have already really thoroughly analyzed single cell data, really thoroughly annotated what the different cell states and cell types are, and have provided them to other people so that they can take the data and almost essentially take our own data and then map it onto these atlases to get an idea of, okay, our cells in this cluster are very similar to their cells in this cluster, or similar to their cells in this case in this cluster. Therefore, they're likely of the same cell state or cell type. So to give you an idea of what an atlas would look like, this is an example of an atlas where they already provided the UMAP, which is just a dimension of lower dimension of the data set that we're actually going to go over in the rest of this data. And so they've already annotated everything so that we can take our data set and map it and see where it clusters similarly. The second way, though, is let's say that you don't have an atlas that you feel really confident in that would be very reflective of the cell states that you're expecting in yours, or you have certain clusters that are not filling well into a certain already defined cell state. And so you're still trying to figure out what they are, and you can't just rely on an atlas. In this case, that's where we want to use marker genes. So we have our clustered cells, and what we can do is, like Jesse already showed, is cluster them and then identify which genes are really defining this cluster as being unique compared to the other clusters. And so what we can then do is we can have databases that we curate ourselves of different cell states that we think are going to be applicable, or we can use databases where they've already done this for us, where they have certain cell types and no marker genes, and then the non-marker genes are those that are not usually expressed in that cell type. And using this information, we can essentially, again, map onto these cell states and see which of these clusters is showing really similar expression patterns to what we would expect from already defined cell states. So with that, let's go ahead and get into actual code of how we actually do this. And I'm going to be showing you on a Jupyter notebook, but you guys are going to have basically the identical code, just in a slightly different format. So first we load up the libraries that we need. And first we're going to go over using a known atlas. So in this case, I label out some resources that you can find for your own single cell analysis of really good atlases to use, as well as a note for loading the data. But in this case, for demonstration purposes, we're actually going to use that data set that already exists, that Jesse and you guys did earlier, but because we have the actual atlas from which that data originates, we're actually going to be able to use their UMAP instead and their clustering to be able to see how accurate these tools can map and go ahead and identify these clusters if we already know what they are. And so in this case, I just loaded one of the Seurat objects that's only looking at a subset of the data because that makes our lives a little bit easier. And then in this case, I'm also going to be splitting it in half so that I can get a query set and an atlas set or a reference set. So essentially, we're going to get what we'd be looking at if we were trying to cluster these cells and identify the cell types on our own. but we're also going to have the atlas that's coming from the exact same data set, and hypothetically, we'll have the exact same cell states and just be the other half of the cells. So basically, we're giving it the most optimal, easiest task that it can be doing. So I'm randomly splitting the data set where each cell state is exactly maintained. We just keep half of it. And I'm showing by graphing the view maps that you can see that these are the two data sets. They look almost identical. We're just taking half of the data set for each of them. So this can be our query set, query, and then this will be our reference set, or the same thing as an atlas. So with that, we're going to use Seurat's inside tool to be able to map our query set to this atlas set to identify cell states. So Seurat has an awesome tutorial that goes into more detail about some other code that you can use, but we're just going to look at the essential basics here, where the first step you need to be able to do is find transfer anchors. So we have these two completely different data sets, and we need to figure out, okay, how can we essentially map one onto the other? And the first step of that is being able to get those anchors. So anchors are essentially pairs of cells from both the reference and then the query data sets that are mutual nearest neighbors, meaning they're very, very similar. And so it's a lot easier to adapt and figure out where other cells that might be a little bit more questioning of how they relate to the reference set when we have a few key pairs of cells that we know are very, very similar. And we can do this by looking at dimension reductions and doing nearest neighbors analyses, but you don't need to do any of that. Just understanding how it does it is good. So you find the transfer anchors, it will get that lower dimensionality, project the query set onto the reference set, find the nearest neighbors, and therefore find the anchors. So we see that we find plenty of anchors to connect them to. And then after we find these anchors, now we can transfer the data labels of interest from the reference set to the query data. And so this is able to use those anchors that we found to compare the individual cells and transfer labels accordingly. So that's where this transfer data is occurring. And then in this case, what we are then doing is just taking that predicted ID from the predictions and putting it onto the metadata. So when we actually look at these predictions that Seurat is getting us, it looks at every single cell in the rows. And for every single cell state that's in that reference, it will essentially score it and see how likely do we think that it falls into that cell state. And so in this case, it will look at the highest score, and that's where it puts the predicted ID. So both of these cells, the highest score was in erythroid, so that's where it's going to map it to. And so that predicted ID is what we're adding to the metadata. And then we could look at that predicted ID and see that this is very similar to the cell labels that we know are true for the broad cell labels. And so because we have that actual truth set, we're actually able to see how well we did. So in this case, I'm just seeing where in which places does the predicted ID match the true ID? And obviously, if you're using a real query set where you don't know the truth, you can't do this. But this is meant to give you an idea of when we have the best case scenario, how well are we doing? So as you can see, we're not perfect, but we're doing pretty well. And then what you guys are going to do in class is going to be repeating the same process, but for the smaller scale cell labels. So we can have our large cell types that we're really, really confident in. Think basic immune cells, so like B cells versus T cells. But we want to get more specific, right? And so we want to consider, okay, which macrophages, what are the macrophages doing? And that's where these smaller clusters and these smaller scale cell states are going to be. So we want to see how well does it perform on these smaller scale cell states. And I've already done it for you below in the code, you just can't see it. And so when I see how well it did, I can see a very similar results where this is the smaller scale predicted ID and then the cell labels, and we can see that those guys did pretty well. And we can compare how well did the smaller scale cell states and the larger scale ones do. And then in this case, I have some reflection questions for you where if you're able to answer these questions, you should have a pretty confident understanding of what's going on. So that is with an atlas, but let's say that you don't have an atlas or you have some certain cell states that you think are much more nuanced that can't really apply. They're not being assigned very well, or You just have some clusters that you would just like to annotate according to some other compared clusters or some specific genes that you think are really important you want to look at. And that's where these marker genes can come in. And so in this case, we can either use a database that's already been developed or a database that we create of cell types and the genes that should go with them. And SC-type is a relatively newer tool that actually does all this work for you, and it's great. And so you can learn more about it. But in this case, we're just going to use one version of it. And as you can see, it already allows you to just automatically load the functions that you need. And then the next step, though, is to be able to bring in that database file. So like I said, you can create your own, but in this case, we're just going to use the one that they already have for the immune system, since our data set fits pretty well into the immune system phenotypes. And so in this case, we're literally pulling an Excel file that they already have that it has in this format. So this gives you an idea of how easily you can do this on your own if you want to, where they have the tissue type. So in this case, we're using immune system. They have the cell type name. In this case, I'm showing the example with platelets. And then they have the list of genes that you would expect to be highly expressed in the cell state, and then the list of genes that you would expect to be lowly expressed in the cell state. So that's literally all of this Excel file is showing. And so they have a function that allows us to automatically load it and then filter it for the tissue. If you see warnings like this, you can see that it's just due to semantics, and it says that everything is corrected, so we don't need to be worried about it. And so when you scroll down, basically we have now this default database, and we can see the different cell types that they're considering. And so these are going to be the cell states that we're going to be able to map our query set to. And so next we need to prep our single cell data. So in this case, all we need to do is that they like to use scaled data. That's what they suggest. And the Seurat object already automatically has that in the process. So this is just capturing that scaled data, and it will figure out whether or not where it is according to your Seurat version already. And then finally, we run single SC type. So what SC type is doing, very similar to before, is it is taking every single cell and it's going to give it a score for each of those predicted clusters. And it's doing that by taking your data. We tell it that it's scaled, and it's taking those marker genes and the non-marker genes, and it's going to score every single cell. And this is what's going to take the longest. And then the fourth step is similar to what we did before. So now that we actually have the single cell data, we need to assign at the cluster level. So in this case, this is just a function that they want us to create. So in this case, we take the clusters of interest, where we're just going to use the broad scale cell types, but you can replace this with Seurat clusters. And everywhere you see this, you would just replace with Seurat clusters, and you could do it on your own query set. And what it's doing is for every single cluster, it's getting the highest score cell type for the cells in that cluster to be able to identify which cell type is most likely going to be explaining that cluster. And so this is actually running our function on each of our clusters. And then finally, they suggest that if a cell state has a score that is lower than the number of cells divided by 4, that's not as high confidence. So in this case, I'm just labeling confidence as being low if that is the case. And so we can finally just look at results, and we can see that we have low confidence here, but we can see, because we know the truth, that it's accurate. And so in this case, we read this as the clusters being our identified clusters that we're giving it. So in this case, we have the names, but if you're using Seurat clusters, it's just going to be numbers. And then the cell state they're connecting it to, the scores, the number of cells that it's considering, and then the confidence level. And so we can see here that it did pretty well. There are some things that don't make too much sense, but still it did pretty well. And so to give you an idea, I just am graphing it so that I'm using certain colors that are coordinating to the different predicted cell types. And as you can see, it did relatively well if we're looking just color-wise. And so then it's your turn to be able to repeat this analysis, but with the smaller scale clusters. And again, I've already run the code to do this, but you're going to type in the code yourself. And then here I can again run a similar cell type or UMAP and show the different comparisons where we see relatively similar results in terms of the number of cell states and similar separations, but not identical. And then we can actually look at the results and compare them and see how well they did. And so again, you have reflection questions here, and I want you to be able to answer these of, were these fine scaled enough to be able to connect them given the database we used? And if you wanted to use a less or more fine scaled database, how might you do that? And once you're able to answer these reflection questions, you should have a pretty good understanding about cell type annotation.